Hi everyone, welcome. So today we're going to be covering literature research tools that are helpful both for teams as they work on projects in the BioBuilder Club and in general for your academic careers and interest in going into the field of STEM. So there's a lot of resources we're going to cover today, so a lot of links, and I will email out a document with all of these resources concisely so that you can use whichever resources you thought sounded helpful. Um, and of course, I will record this meeting and send that recording out to teams as well, so you can always watch it back later. I'm still letting people into the Zoom as they come. Okay, so welcome everyone. This is kind of an overview of the topics that we will be covering today. Just this here. This is an overview of the topics we'll be covering today. And it is a lot, but a lot of it will be summarized in both the BioBuilder Club handbook section on literature research and also in the review follow-up email from this meeting. So before we kind of dive into reading primary scientific literature, I wanted to share some more general resources that your teams can use to learn about synthetic biology and get some project idea inspiration. I'm also going to talk about our own project library for the BioBuilder Club, as well as how to access the iGEM project library. Then we're going to go over some general pointers about reading scientific literature, some resources that can be helpful in learning about the different parts of how articles are structured. And then one approach to reading papers, which I think works really well for a lot of students, called 10 Simple Rules for Reading a Scientific Paper. And we actually only need to cover the first 5. And then I'm going to talk about how to access articles for free, which I know a lot of people are here tonight to find out how do you get access to articles for free. So we will cover using Google Scholar and PubMed and some other methods to access free articles, and also how to email authors. So it's very common in the scientific community. If you need a paper or if you have questions about a paper, you can reach out to the people who wrote it. And journals are set up to provide this information. And so we'll talk a little bit about that as well. Also, I know that a lot of schools have different policies when it comes to using AI, whether or not you're allowed to access AI tools at all at school. But I think AI is here, and I have some resources that you can use to evaluate different AI tools and suggest some free ones that are designed specifically to help researchers that might be useful in your literature research. I'm also going to talk a little bit about citation management and tools that are really useful for online-based collaboration these days. And then I do have a couple more tips on ways to approach reading articles as a team or as a class. So in terms of my general SynBio resources, all of these links here are for different sites. Ginkgo is a biotech company. One of our learning labs is actually located in the Ginkgo headquarters in the Seaport District of Boston. And they have an interesting blog called Grow by Ginkgo. There's some podcasts here. And Symbio Beta is an industry organization. So all of these different sites will have a lot of examples of what's happening right now in synthetic biology, directions the field might be going in. So you can kind of get a broad sense of the various applications. When it comes to kind of finding a topic of interest for your team, you can start out, are you interested in environmental issues? We'll look at some of these sites for projects that are on any issue, whether it's local or global. If you want to work on a local issue, you can also refer to local sources. You can even see what local researchers, even if it's not necessarily SynBio, are working on different issues if you're interested in the environment, and then see if you can take a SynBio approach to that. You can also hop on the BioBuilder Discord server and discuss your ideas and ask for suggestions from other teachers, students, mentors who are on our server. And I will again share the link for joining our server so that I think a lot of people are on it now, but just to make sure. So all of the past BioBuilder Club projects are in our project library, which has been revamped this year. It's really pretty. And you can go through and search by year and school and topic and see the presentations, the slides, the audio for the talks of past teams. So that's a good resource if you want to kind of pick up a project that's been worked on in the past a bit, or if you just kind of want to see what are the types of projects and their scope the other teams have. We also, I would suggest checking out the iGEM project library. So I know many of you are familiar with iGEM, but it's the International Genetically Engineered Machines competition, and it's the world's largest symbio competition. And they have both an undergraduate division and a high school division. Some of our BioBuilder Club teams participate in iGEM. And these projects all are accessible on the iGEM project library. Each team has a wiki, and you can look up the details of their project and who was on their team, as well as what awards and things they received at the end of their season at their jamboree, which just happened in Paris. So those are really good resources. iGEM has the different village categories. I forget how many there are now. There's like 15 or so. And those are different topics. And when you request your mentors on the form, I do ask teams those questions. Are you interested in bioremediation? Are you interested in, medical diagnostics or therapeutics? So those project libraries are both really good resources. And if anyone has questions, feel free to interrupt me or put them in the chat. Another really good resource specifically for high school science, synthetic biology. Sorry, I'm having issues with my zooming here. So another resource that's really good specifically for high school symbio is BioTrex. So I know a lot of teams are familiar with this. It's the peer-reviewed journal for high school synthetic biology. So at the end of March, BioTrex is open for article submissions, and many BioBuilder Club teams, I think 35 this year, are going to be publishing. And so the peer review process starts when you submit your abstract. And we're going to have some BioBuilder Club teams later in the season talk about their experiences with publishing in BioTrex. But you can publish several different articles. If you go to their website, there's a for authors section, and it'll talk about the different manuscripts that they accept. But briefly, you can publish design-only articles. You can publish methods, perspectives, and reports. So 35 teams are publishing in the edition that's coming out anytime now. And BioTrex is open, though, to any high school students. It's not exclusively the BioBuilder Club or iGEM. And this is also a really great place to see the kinds of projects that students in high schools around the world have worked on. And you can also find out students did the research and what schools and how you might be able to contact them. And so BioTrex is another good place to look for project inspiration. So those are my kind of more general synthetic biology resources. And now we can dive into what everyone is here for on Literature Research Night, the literature. So when people say, the literature, they're usually talking about primary research literature. So these are peer-reviewed articles that are reports of new research on a specific question. Now, not all peer-reviewed articles are primary literature research. Some peer-review articles are actually review articles. Those are not presenting a report of new research. Those are summarizing other primary research articles to kind of give you a snapshot of that field and a particular question at that moment. So when you do come across peer-reviewed journals, you'll need to make sure you understand what article type you're reading. So primary research literature is giving you new research report, and peer reviewed is giving you a summary. So you'll have to be especially careful if you're using review articles that you look at when they were published, because a lot of fields have changed in the last decade. And so you'll want to make sure that if you're using these review articles, that you are using, in fact, pretty current review articles. So um, now that we have talked about what exactly is the literature, it's important to note that reading scientific papers is not like reading newspaper articles. So a lot of those resources that I shared in the early slides are for a more general audience. So those articles would be writing about science, but peer-reviewed primary research literature or review articles are very different when they are the science. And I'm going to share some strategies because unlike a regular article in a magazine or newspaper, you don't necessarily just start at the top and finish at the bottom and move on with your day. You have to be very strategic about why you're reading the article, and that can inform what sections you read the article in, how you take notes, what you're going to reread, what references or citations you're going to look up. And another note is that just because it's a peer-reviewed journal doesn't mean that they're all equivalent. And so there's a handy resource from the Web of Science. It has indexed lots of scientific journals, and so you can get a sense of which ones meet criteria for being considered more reliable and having a higher standard of evidence than some other ones. So reading research articles is a skill you definitely have to practice. And everyone in the BioBuilder Club is at an advantage here because you're practicing this skill of reading primary research literature starting in high school, which is amazing. Because a lot of people don't get to it until undergrad or even grad school. So in terms of where to kind of orient yourself, if this is your first journey into reading scientific literature, you might want to check out some of these resources. The anatomy of a scholarly article is pretty cool from the North Carolina State University Library. And you can go into it, and it has research articles, and it has annotated the different parts, and you can click on it and find out what to expect in everything from the title to the author list to the formatting of the methods and results and citations. There's a really cool resource called Dissect an Article from a university or Arizona State University library resource. And it helps students practice reading biology articles. So it uses articles from PLOS, and it has kind of dissected them and annotated them. And this is a good way to practice how to interpret the different sections of articles. And if you prefer learning from watching and listening, you can check out this How to Read and Comprehend a Scientific Research article. It's only 5 minutes from the University of Minnesota Libraries. So once you've looked at some of these resources, you're kind of oriented as to how these articles are formatted. And so once you have some experience with reading articles, if you want to see something very entertaining, in 2016, Adam Rubin published a very tongue-in-cheek How to Read a Scientific Paper. Mentors and teachers on the call might especially like reading that one. And then in response, Elizabeth Payne published How to Seriously Read a Scientific Paper. And this is a cool resource because she interviews a dozen scientists or so from different fields of life science about their and different career stages about their strategies for reading papers. And I guess I am sharing this here because there's not just one way to read a paper, just as like there's not one way to do synthetic biology. And so it's really going to depend on your preferences, your learning style, and your goal for reading that particular paper. So you don't approach all papers with the same goal. So that brings us 10 simple rules for reading a scientific paper. So this was published in the PLOS Journal for Computational Biology. I think it applies really well to a lot of fields of science, and especially life science. So I've only summarized here rules 1 through 5. because those are kind of the big picture recommendations for the steps you should take. And I think that they are the heart of the how to read a scientific paper. If you want to read the rest, I will share that link so you can read rules 6 through 10. So the first thing to keep in mind when approaching a scientific paper is that what is your goal in reading this? So maybe you're new to reading a scientific paper. Well, in that case, you should probably pay particular attention to the figures and start to learn how to interpret that. That is where the data is communicated. Maybe you're entering a new field and you want to learn like what is important in this field right now. Well, then you might be particularly interested in the introduction, where it lays out the motivation for this research, and the discussion or conclusion, where it talks about next steps. In the full linked article for here, it lists a lot of other reading goals that are applicable to different career stages. So it's definitely an interesting resource to continue to refer back to. So once you have your goal, rule number 2 is to understand the author's goal. So this requires a little bit of background work. And it's important to keep in mind that sometimes an author will work on research and it will be published, but it might not necessarily be their specific field. It might be adjacent to it. And so kind of learning that context of who wrote the article and where is very helpful. At this stage, you also have to consider the type of articles. So we mentioned primary research articles, we mentioned review articles. There are also articles that focus on methods or commentary in response to an article that was published in the previous edition of that journal. So each one of those has a standardized format, and they're usually identified in the article type when you look at the journal. And we'll look at a couple of those. So the next step here is that you want to ask 6 questions. Now, these 6 questions, it's sort of cheating because rule 3 is actually 6 questions. But these 6 questions apply both to the full article in its entirety, and also if you're looking at individual figures within the article. So you just want to know, what did the authors want to know? What did they actually do? Why did they do it that way? What do the results show? What do the authors say the results show? And then what should be done next? And do you agree with their interpretation? So that will come up again later, those rules. And then for us, we want to make sure that we are really unpacking each figure and table. I think a huge difference between scientific articles and regular articles is that sometimes you don't miss much if you're reading an article in a magazine and you don't look at the pictures. But in a scientific article, if we're going to do anything, figures are super, super important. And you really want to unpack each of those figures and tables. And at the end of the presentation, I'll talk about a few reading strategies for groups. And they have to do with assigning each person a figure that they're responsible for unpacking and sharing with the group. So in original research papers, the data is the important part. That is why they're publishing this. So some people like to kind of summarize the figures and tables before reading other text, because you really should be able to get all of that from the data itself. And for each figure, scientific figures are incredibly dense. I think that that's one of the challenging things about getting used to reading them. It matters. You got to understand what all of the axes mean. The color schemes are not random. You have to understand the statistical approaches that the authors chose. Why is the figure structured this way? And the legend is important. You do have to read the legend. That typically will kind of give a summary of what this figure is about, how they got that data. But when you're digging through what this figure means, you might also have to look in the other sections, like the methods, to find out what exactly is done. So you can go back to that rule number 3 with my 6 questions and look at that about each panel in the figure. Basically, those 6 questions are just a really great way to interrogate any part of someone else's data that you're looking at. And then what is the take-home message of that figure? So the last rule that we're going to go over is Rule 5, understanding the formatting intentions. And this might be relevant before the step 5, but it's important to understand the different parts of the article. And I will mention here before we dig into finding research articles, the author list depends the standards based on the field of research, but in biomedical research, for the majority of cases, when there's an author list, the first author listed, it's called the first author, is the one who was in charge of that project's implementation. These are often graduate students whose work is presented. And then the last author in the list of authors is going to be the person who supervised that project. So the principal investigator of that laboratory where the research was conducted. Of course, sometimes this gets slightly more complicated when there's collaborations between different labs and larger institutions. But for the most part, first author really led the implementation of the project. And the last author is the one who was the primary supervisor. And so when we are looking for contact information that the journal will provide, a lot of times that contact information will be for that last author, but it's also possible to find out the email address of that first author as well and reach out to them directly. And then research articles, another difference compared to just regular articles is that they have an abstract. So the abstract is basically the elevator pitch for that article. The other parts of an article is the introduction. So this presents the research question, why it's worth writing a paper about. And from reading the introduction, it should give you the context, what has happened leading up to the research that this author is presenting. And then in the methods section, how was the research that they're presenting done? And then in the results, they're going to be communicating kind of objective observations, should be objective observations about what they found. And then finally, the figures, like we talked about, should kind of convey all of that data from this research. And then in the discussion section, and you might see it called discussion, conclusions, a couple other terms based on the journal, that's the author's interpretation of their results. So in the introduction, they told you about the context. The discussion should show how this fits within that context and what they think it means for the field and directions moving forward. So that's kind of like the crash course in how scientific articles are written. So now, how do we find them? One of the resources that I like to recommend first to high school students, because it's easily accessible and free, is Google Scholar. So a couple things before you necessarily jump right into the primary literature, it can sometimes be helpful to pick up a secondary source. So those are sources that are summarizing this research. So even things like the Wikipedia articles, for example, a lot of teens are interested in microplastics. You can check out the Wikipedia article on microplastics, and it might show you terms like microbial biodegradation and bioremediation. And then you might realize that you'll want to use those search terms when you're looking for primary research literature on microplastics. So if you find a paper, even if it doesn't have what you want, it can still be very useful. So even if the search results are more specific than you need them to be, you can look in the references section. So probably in the introduction to a paper that's more specific than you need, they're going to give you context on the field, and they're going to cite papers, including review articles that you can then go to that might kind of zoom back out to a more general level than that paper. So for example, you might find a paper that's really into the methods and you just kind of want more of that context. So what are they citing in the introduction? Can you find any review articles to give you a sense of the state of the field before you dive into that primary research literature? And if you find that the articles that you're pulling up are too general, you're looking for something a whole lot more specific than this kind of general overview of the field, on sites like Google Scholar and other tools that we're about to discuss, there's going to be a cited by section. So somewhere on like a sidebar at the bottom of the journal page, there's going to be cited by, and then it'll list and be hyperlinked to articles that have referenced what you're currently reading. So those articles, because they're citing as background information what you're reading, are going to be generally more specific in nature. You can also check out related articles on Google Scholar. This is an option to see works that are closely related. And actually, we're going to talk about some AI tools that are also designed to kind of bring out these connections and relationships between different research and kind of pointing you in the direction of research you're probably going to be interested in based on the history of what you've been looking. So within Google Scholar, there's a couple of options. On the main page, there's like an articles radio button field thing, and then like a case law. You probably don't need to look up case law for your BioBuilder Club projects. So Google Scholar is not specific to biomedical research. So you're going to find a lot on it that pertains to other fields. So sometimes when you're using Google Scholar, you're going to have to pay attention to some of the more advanced features to get the most out of it. When you pull up an article and you see one that you're interested by, it'll have the title and the authors and the beginning of the abstract. And then underneath it, there's kind of a menu bar. You can see how many articles it's been cited by. You can save it, so you can have a collection of articles that you've saved in Google Scholar. You can get a citation in the format you want through the cite button. And most articles are available on the internet in multiple versions. So one approach to trying to find those free articles has to do with looking at the different versions. Whenever you see this next to a search result in Google Scholar when it says PDF, that means that there's a PDF here that you can download for free and access that article. Other important features of Google Scholar is when you save your articles here, they'll go into this My Library, which you can then open up and organize and share articles with other people. You can also click here under articles on profiles. And so certain researchers who are prominent in a field and have published a lot in that field, you can get more information about them specifically. You can also just Google professors and principal investigators and find their university pages that they're affiliated with. If you're really into a topic and you want the most cutting edge research in it, you can sign up for Google Alerts so that when something is published with certain keywords, you will get an email. There are also advanced search features if you want to say you're only looking for articles since 2023 or something like that, or if you only want like a certain type of article. So Google Scholar is a good option for a lot of general research. It's good even outside of BioBuilderClub projects if you're looking at other fields of science. Questions so far on all of those before I dive into PubMed? Okay, PubMed it is. So specific for biomedical research is PubMed. So this is a yes, Rick, I totally recommend using the citation manager, and I think in 2 slides I talk about those. A lot of the citation managers that people working in industry or academia have access to through institutional affiliations are not available to students, but I have a list of so I'll definitely be going over those. And my tagline for citation managers is that anyone you use is better than not using one. But yes, I'll talk about those in a sec. So PubMed is a free resource, and it has, it's probably a lot more now. This is an old figure, 36 million citations and abstracts from specifically biomedical literature. A lot of times you can also find the full text links, kind of like in Google Scholar. Sometimes there'll be like a box with the journal's logo, or it says like free full text, and you can click and get it there. You can also look in PubMed Central. So PubMed Central has free full text articles that are also categorized in PubMed. So within PubMed on the left side of the panel, when you're using a web browser, there's a lot of filters which you can change if you only want freely available articles, if you want to see only review articles, if you want to restrict the publication date. There's also tags you can put if you only want articles within a specific field to show on your search results, or if you want to get really crazy with some Boolean operators, which is totally possible. A really kind of cool feature, I think, of PubMed is that you can actually visualize the results by year. So if you want to see what year a specific field that you're interested in really took off, you can see when there's that spike in number of publications in PubMed. So PubMed searches are really useful. It automatically uses term mapping to help identify concepts. But increasingly, people are also using AI tools to kind of replace some of these advanced search features. But I still recommend checking out how to do those things with PubMed and Google Scholar, especially since many schools, high schools, do have, I know, policies where you're prohibited from using AI. So if you take one thing home from this presentation, don't pay for scientific articles. So there's 4 steps. One is a lot of times you can access the same article for free from one source, but not another. So that's why sites like Google Scholar or even just regular Google are really good. Just type in, you want the free or the full text with PDF. And so it's the same exact article, but one source makes it freely available, the PDF for download, and the other would charge you $34.99 or whatever. Don't pay that. Number 2 is that everyone, I think for the most part in the BioBuilder Club, is associated with some kind of school. And a lot of times your schools or even local libraries will have subscriptions to some databases. Obviously, universities have subscriptions to a lot more databases, but it is always worth checking with your school and local librarians to see if you can log into certain databases that otherwise people would have to pay for these articles. Another step is when you're working with a mentor in the BioBuilder Club, sometimes your mentor can download the articles for you if they're behind a paywall, if they have institutional access through their affiliated workplaces or universities. And then if all else fails, you can email the authors. I know that a lot of students are maybe hesitant about emailing researchers, but honestly, they love to hear from students. If you're very polite, they will send you their article. And if you are very polite and have questions and you're concise and to the point, they might even help answer some of your questions if you haven't you've read the paper. They'll probably want you to read their work, and they have PDF copies of it, so they are allowed to distribute those. So when you are using any article and emailing the author, I'll show you right here, it will show you probably the corresponding author's email. So the corresponding author is sometimes the last author. This article, A Global Perspective on Microplastics, the first author, Robert C. Hale, is actually the corresponding author. And so it gives an email, and you can email him, and he would probably give you the PDF of this article, although this one is available for download. Sometimes the corresponding author is the last author or the primary supervisor of the project. Sometimes if you email them, they'll just send you the article. Sometimes they might put you in touch with a graduate student. And other times, if you don't hear back from them, it's worth Googling the first author and seeing what institution they're at now, and if you can get their email and email them as well. Google Scholar as a great resource for getting free full articles. If you're logged into either like your school library or you have institutional access, a lot of times there'll be a link through your library where it says something like find it at Harvard. And then what that means is that on the back end, using the institutional subscriptions your organization has, it will find what source has the article that it is subscribed to so that you can download it. Anytime it says PDF or even HTML to the right of the page, you can get that. So some of these are what's considered preprints. It's the same article, but sometimes there's the free PDF version available before it's finalized, like the formatting for publication. And then if you can't get that article, you can also click on related articles or to explore what cites that article. But if there is a particular article that you really, really, really want, you can't get access to it, definitely you can email the authors. And if you really, really can't find it, you could even email. If you can't get it through your mentors either, you could try reaching out to me and I might be able to get it. Okay, so any questions on that before we talk about some AI tools, citation management, and research? Okay, so there's been an explosion in AI tools, everybody, but there's a lot of them geared specifically for researchers and conducting literature research. So the Purdue Library Guide has a very nice overview on AI tools in research, the limitation of those tools, and using AI for systematic review. There are a lot of different AI tools out there, and they each have intended purposes that they work better for than others. You do have to be careful because with generative AI, it is possible, like if you're just asking ChatGPT questions, that it will make up sources. So you need to make sure that if you're trying to conduct literature search and find actual papers, that you're using an AI tool that uses citations that exist. Because obviously you're not going to be able to find a PDF version of an article that ChatGPT made up. So Georgetown University has a good guide to these generative AI resources. And I've included on the next slide the ones that are either entirely free or have free versions that I think are worthwhile checking out for different purposes. There's also an organization that has this generative AI product tracker because there's so many tools these days. And so that kind of synthesizes their primary purposes, their limitations, and whether you can get them for free or not. So AI tools that do have free versions, I kind of organized into three groups. Some tools are for visualizing citation networks. So these will actually generate like citation maps and citation charts. So you can see how papers are connected. One of those tools is literally called Connected Papers. And with that, you kind of give it one seed paper to start with, and it's kind of like a mind map fit that it generates. Lit Maps is very similar, but you can actually upload multiple articles as a starting point. So it starts out with a little bit more complex picture of what field you're looking at. I really like Research Rabbit because it actually is free. It's not just a free version, it's just free. And this is a citation mapping tool that kind of lets you focus on the relationships between those works, those visualizations like the previous two tools, and it can also help suggest literature that you haven't read yet. Another category of AI research tools are those that are kind of designed to generate article recommendations. So QDS and Insightful both have free versions or like limited free versions. And one of them is kind of like a recommendation search engine based on papers that you upload. And the other one is interesting because you can upload two papers and it will show you kind of how they're connected through a web of citations. AI tools that go in a little bit of a different direction include those that actually are kind of synthesizing research findings. So one of those is called Consensus, and it uses large language models, and it will kind of synthesize research, and then you can ask it questions, and it will answer based on the research, and it does provide citations that are not made up, so that's helpful. So you can then actually go track down those papers and read them. Undermined is like designed to be like an AI research assistant. So using literature and these citation connections, it can kind of help you refine your research question and then find the relevant literature. So with the exception of Research Rabbit, I think most of these tools have a free version, but the free version obviously has limitations in terms of what features you can use compared to the paid versions. And they also might set a limit on like the number of search queries you're able to use in a certain period of time. But I know a lot of BioBuilder Club teams have used Research Rabbit. On Discord, if anyone has other suggestions or if there's a tool that your team is particularly passionate about, by all means, please share those. These are just the tools that I am currently most aware of here. I will also say that a lot of these AI tools rely on some of the same underlying data sets. So Semantic Scholar is a really cool resource, and a lot of those on the previous page use the database from Semantic Scholar. And it is free, and it's this AI-powered research tool. It has indexed over 200 million academic papers. That's kind of the data set that the other tools are using. And it's cool because it's free and you can kind of get these TLDRs from the papers. Another data set that's really commonly used is from OpenAlex. There's a similar number of indexed papers, but interestingly, it has more coverage of other fields which might not be as relevant, like humanities, but it also indexes journals from non-English languages with a focus on the global South. So that's really cool. If you're interested in kind of certain research topics that are affecting different areas in terms of climate change and things like that, you can check out Alex. Citation management is also, I think, a really, really useful tool. It's what I was asking about. So these help keep track of what you read. It allows you to not just save the citations, but a lot of them you can actually save the PDFs, the articles. You can tag them, you can annotate them, and then you can export these for bibliographies and sources, and you can share them with others. Like I said earlier, I totally agree with the premise that using any citation manager is better than using none at all. When you're working with primary scientific research literature, it can get out of hand the number of papers that you're ending up juggling. And especially because you're all working on group projects, you want to make sure that you stay as organized as possible, and that you know what articles you're referring to when you're communicating with each other, with your mentors, with your teacher. And so there's actually here, and I'll share it in the follow-up, tips on collaborating with citation managers, because depending on which one you're using, collaborating is a little bit different. And so some of the citation management programs, you can kind of work together on a project, but each annotate them differently. So definitely check into those. I think when teams ask me about what citation manager to use, I usually say Zotero because it's open source. But there are basic plans or limited plans of Mendeley, EndNote, and then high schools sometimes have subscriptions to something called Noodle Tools. I don't know if any of your schools do, but it's possible. or Power Notes. And so there's kind of 3 main parts of a citation management system. One is that it collects your citations and lets you also save actual articles to it. 2, most of them have a browser plugin, so that way you can actually really easily add the citations to the management system, and it's not a lot of work to put them in. And then 3, most of them have some way to export the citation in whatever style you choose, so that you can collaborate with your team and with your mentor. When you're working with mentors, I think that an essential part of kind of the research for your project is conducting a literature review and being able to provide those sources in an organized, annotated way so that you can ask questions about the research to your mentor and they can answer them. I highly recommend a citation management system. So use any one, just don't use none. Okay, any questions on that or recommendations for other citation management systems that you might be able to use for free that I didn't mention? I don't know if anyone's.
Chloe?
Yeah.
So are the students using APA style citations?
So I personally am not super picky on how they want to go about this. But I will put citation styles and recommendations in the follow-up email so that you can see, and I will put like how to get that from like Zotero and other things in there. But yeah, our emphasis is that I would rather things be cited than not cited. I will also say, even though the final assembly and projects are ways off, posters are only so big, and this is a problem because we want teams to be able to reflect the whole breadth of research that their project is premised upon. So a lot of teams will make a citation document that's available by QR code on their posters. That way you can look up those references and also have them highlighted and things like that. So don't feel like you have to constrain yourself to the number of resources that could fit in the corner of a poster. So that's just something kind of to think in mind. And also just kind of get thinking about the fact that you really should use a citation manager and start annotating these things so that way when it does come to preparing your posters and presentations, all of that is set and ready to go. And when you're writing about your projects and your introductions, you're not having to figure out like going backwards trying to find out what to cite. So I highly, highly recommend trying to stay as organized as possible with this and use some kind of collaborative citation manager or some collaborative document like a Google Doc. I don't know if experienced students and teachers on the call have any suggestions, but I know a lot of teams use Google Docs for this to communicate with their mentors, but it's definitely up to the team. Up there, Anna shared her teams use the My Bibliography tool.
It's a APA generator. You just put in the link and it'll generate for the most part, it's about 90-80% right. You might have to do it manually, but it automatically generates the APA 7 style in there.
And well, for the most part, if you're using like articles, you on most journals or through like PubMed, you're actually able to download citations in whatever format you want. There's usually a few options. A lot of things on PubMed have like a button you can click that's called Cite This, and then you pick your citation style. But yeah, so Anna's MyBib resource that she shared is really good. So if that's not an option for the articles, that way students don't have to be looking through the reference books to figure out how to cite things. But yeah, my bib is free, so that's a good resource too. And a lot of times though, well, on Google Scholar, it can be tricky. Sometimes if you just download the PDF, it doesn't have an option for you to export the citation. But if you're using a citation manager, a lot of times that can extract the citation from whatever you're looking at, even if it doesn't have a cite this button. So highly recommend citation managers. Anna, does your team use any of them like Zotero or anything?
No, I don't know anything about them. What I do is I made a document and then they put the article there, they put what they got out of the article, and then they put the APA 7 style, and I put the generator up there so they automatically, cite it that way. And then they can go back and
That's
figure
great.
it out
Yeah.
that way. Yeah. But yeah, send me I would love the
Yeah,
information.
I recommend it. Citation managers are great.
Okay.
So yeah, that can definitely, it's kind of like exactly what you're talking about. Like it has the citation saved, it has the article, and then you can annotate it, annotate that citation. But yeah, I'll share that because that's really easy. But yeah, definitely a manual way to do that is just using Google Docs. That's awesome. So I have received a lot of questions from teachers specifically, but this is definitely important for students too, on how, as a class or a team, to read a research article. And one of my favorite approaches that I have come across is the jigsaw approach. Now, you might have heard this in the context of, like, I don't know, reading other articles or novels in any kind of academic subject, where you just split it apart and you make different groups or individuals responsible for different parts of whatever you're reading. But I like this specifically applied to articles where everyone is responsible for reading the introduction. You've got to understand the background and context of the article. And then if you go back to my 10 simple rules that someone else wrote that I really like, rule number 2 was understand the author's goal. So everyone should be able to accomplish rule number 2. Then you split up the figures, giving 1 or 2 figures to each student or small group of students, depending on how many students are working on this. And that's their figure to explain to the larger group. They're not just going to be like, this is what's in the figure. They have to actually really break that down to figure out the research and interpretation involved in the presentation of that figure in the paper. So in terms of the 10 simple rules, that would be rule number 3. So those 6 questions that you can ask of an article as a whole, you can ask of an individual figure. Some papers are like ridiculously dense. Sometimes you'll even break it down and instead of a whole figure, you have a panel from a figure. You can still use those 6 questions on one panel. So I love it. You can use them on a panel, you can use them on a figure, you can use it on the whole article. And you'll also want to refer to rule number 4. So just to remind us, because that was many slides ago, put that on the next slide. And so for the figure that students are responsible for, those 6 questions from rule number 3, this is a good time to make sure that students can answer those for whatever figure they're assigned to. And then rule number 4 applies specifically to figures. So because they're so information dense in biomedical literature, it's important to understand like every detail of a figure. What are the axes? What are the units on the axes? What does the color scheme or color gradient mean? What statistical approach was used to analyze the data? How was the data presented? Why? And then that take-home message. So once students have this for their figure, theoretically, you have covered all of the data in that paper, and then they would present their figure to the larger group and kind of have a group discussion. And then you can use those 6 questions as a group to discuss the entire article as a whole. So that's just one approach. I think that it works pretty well personally. But there's a lot of different ways that you can read this. And again, there's not just one way to read an article. It's not just one way to do this in bio. If you're reading a research article on your own, though, I wanted to kind of give you some suggestions. This is from a Duke Library resource, and it's the like section by section approach, but it doesn't actually have you read the sections in order. So you start again with introduction, and then as you're kind of orienting yourself to a field, so maybe this is one of the first few articles in the field you've read, you want to figure out what's the big questions, like why does this field exist? What's like the big question they're trying to solve? And then you should take a look at the introduction and kind of summarize the background and context very briefly. And then identify the specific question that this article is trying to answer. And then also what approach the authors have taken in this particular paper. And then you would go to the methods section. And this isn't always necessary, because again, you're not always reading papers with the same goal. But if you need to understand the paper in its entirety, it can actually really help if you draw diagrams for the experiments that the authors are describing what they did, so you can kind of visualize that. That can also be kind of interesting when students are learning about different techniques to kind of actually draw that out and label it. Then you go to the results section and summarize each of the experiments, figures, tables. And again, I just like to emphasize the figures and tables in primary research article are that's the heart of it, right? So that's what they're communicating. That's why they're writing. That's why it got published. So don't gloss over them. If you're going to do anything, read the figures, understand the figures. And then kind of you ask yourself some questions. Do the results answer the question that the author set out to answer? What do you think the results mean? What do the authors think they mean? Do you agree with them? Right, so researchers, no matter what career stage or institution or article or what journal it's published it in, you know, research is evolving. People aren't infallible. And so you want to see, did they miss anything? What are next steps you might do that they didn't suggest? Do you agree with them? So then in this approach, you read the abstract. Some people really don't like to read the abstract until the rest of the article, because the abstract is very much laying out the author's interpretation. And some people want to kind of figure out their own before they read the abstract. And then you also might want to see what are experts in this field, if this is just one of the experts, or maybe someone who's not an expert in the field. What do they have to say? Like, what has the discussion been in articles that cite this? And then you can also review the citations to see what else that these authors cited. So those are my tips. I know that there's like a lot of resources in this, so I will send them all out in an easy to read document. And hopefully everyone came away from this presentation with something new in terms of a article reading strategy, or a resource or tool to use. So take home message, read lots of articles, don't buy any of them. Yeah, questions? Comments? Well, was it helpful? You learned something? Yes. Okay. Thank you. Awesome. So those are all the resources I have to share here. If you have any questions or anything that specifically comes up for your team, feel free to reach out to me. I know some students are asking about assessing the video. So those teachers will have another document that I'm emailing to your teachers. And then I will send the follow-up as well. So I will post this on YouTube and distribute the link as well. And I'll post the slides and share those. So thanks, everyone. Hope you have a great night. And I'll see you guys at the next webinar. Bye.
Hey, Chloe.
Oh, yeah.
Hi,
Anna.
I adopted a cat and it keeps going behind my computer, knocking everything down.
Oh,
yeah. So every day, yeah, we're getting better at it. But anyway, so you've spoken with, this is the part that you've spoken with Janet about doing the local one, the local final assembly, a regional final assembly.
yes, so there will be one. Yep, I'm waiting to talk to Janet more about that.
So I'm kind of arranging it, and I found a place, and I have to speak with her about it. We're tentatively, we're doing it on the 26th from 9 to 1.
Okay, awesome. So is it not going to be at Georgia Tech?
No. No,
Okay.
they don't have the place. I got Brookhaven City Hall. It's really it's brand new. It's really
Oh,
great. So in the main level, we're going to have the poster session, and then they have the chamber, which will be great for a lightning talk.
awesome. So you said
Um,
March 26th from 10 to 1.
well, I put 9 to 1. I got the place from 9 to
9
1.
to
So I'm
okay.
not, sure how many
And
people
sorry,
are
what
gonna
was
be
the
going.
place called again? I didn't catch
It's
the city name.
called Brookhaven. Let me put it in the thing. It's called Brookhaven City Hall, and it's on
Okay.
Peachtree and